Good morning, everyone. My name is Natalie, and I would like to take the next 5 minutes to talk to you about bioremediation. So, what is bioremediation? It is the treatment of pollutants or contaminants using microorganisms, bacteria, fungi, or plants to remove or break down the contaminant into a less harmful form. And it can be used to remove contaminants from soil, water, and waste material. So, next up, I'd like to talk to you about the role of microorganisms in bioremediation. Microorganisms are the foundation of the bioremediation process and do the majority of the hard work. So, first of all, microorganisms produce enzymes that allow them to break down pollutants, and then the microorganisms use the contaminants that they've broken down as a food source. Next, they excrete byproducts such as carbon dioxide and water, and finally, the microorganisms reproduce. So, next, I'm going to talk to you about a couple of the different types of bioremediation. There are many, and I'm just going to focus on 3. So, firstly, in situ bioremediation takes place in the original location of contamination. 3 types of microorganisms can be used. Firstly, microorganisms native to the site can be used. Alternatively, non-native microorganisms can be introduced. And lastly, bioengineered microorganisms can be designed for the site. So the microorganisms chosen then work to break down the contaminant. And this photo on the left shows an example of microorganisms being used to remove oil from a soil medium. So another type of bioremediation is ex situ. This type of bioremediation involves treating contaminated material outside of the original location. An example of this type of bioremediation is the use of bioreactors in wastewater treatment. Bioreactors are seeded with microorganisms, and wastewater is fed into the bioreactor. The bacteria then work to break down the undesirable contaminant, and the water is released from the reactor. There is typically a filtration process associated with this that retains the bacteria and the contaminant. Lastly, I'll talk about phytoremediation. As discussed by Rennie, phytoremediation is a technique which uses plants to remove contaminants from soil or water. For example, you can use giant reeds to remove selenium from marshes or wetlands. And the selenium is then volatilized into the atmosphere, and other contaminants can be metabolized as well. So there are many different applications for bioremediation. Just to name a few, it can be used in industrial wastewater treatment, surface and groundwater remediation, and contaminated soil remediation. When planning a bioremediation project, there are a number of considerations that one must make. Firstly, what is the contaminant and which microorganisms are capable of degrading this contaminant? Next, what are the physical and geochemical properties of the site and which microorganisms do these conditions support? Lastly, what are the byproducts of the reactions that are going to take place, and are these byproducts safe? So I'd like to talk about a case study where bioremediation was used to clean up the oil spill by Exxon Valdez. So 11 million gallons of crude oil were spilled off the Alaskan coastline, and the EPA decided to use bioremediation as part of their cleanup strategy. They determined that there were pre-existing microorganisms in the environment that were capable of degrading oil, and so they decided to enhance the growth of these pre-existing populations using a fertilizer that contained nitrogen and phosphorus. They then applied that fertilizer to beaches and tidal zones, and as you can see from the top photo, the mussels on the left are pre-bioremediation, and they are covered in oil, and the ones on the right are post-bioremediation, a much cleaner mussel. So I'd like to leave you with the pros and cons of bioremediation. Firstly, it can treat contaminants in situ, reducing the need to transport material, and is less disruptive. It is based on natural processes and is typically less expensive than traditional methods. And contaminants are usually removed rather than just moved. The cons are it's generally a slower process and can produce undesirable byproducts, and it tends to be research intensive. So that is all, and I would like to say thank you so much to all of you for listening. Thank you to Dr. Nagati, our TA, my primary reviewers, and my secondary reviewers, and my references are here. Thank you so much.